My name is Charles Liu. I am a professor of neurosurgery, neurology, and biomedical engineering at the University of Southern California. I am the neurosurgeon involved in the project and one of the site PIs here at USC. What we did was we implanted two micro electrode arrays in his brain, and the arrays then would record directly from the firing of the neurons or the cells in the brain. These recordings would be decoded by algorithms developed by Dr. Anderson in his laboratory, and these decoding algorithms would then be used to move a robot arm just with his thoughts. Eric's surgery was done in 2013 in April. The surgery itself took about 5 hours in the operating room. It was a little challenging because Eric, being a high spinal cord injured patient, is a little bit more fragile than our typical patient. Positioning is a bit of a challenge. Because it's the first implant in this area, everything about it was different. In a human being, we've done these types of surgeries in other locations, but when the location changes, the positioning of the patient and how you set up the operation and how you manage all the hardware that you're implanting becomes different as well. Now we have an entirely new area in the brain that can potentially be very useful to achieve the kind of direct brain-machine interface control that opens up an entire new direction in development of technologies, transformative technologies that are designed to give patients who have suffered neurological injuries the capacity to become more independent than they are now. When you think about suffering a catastrophic injury such as a spinal cord injury, the prospects are pretty daunting, and having a new possibility, a new technology that now has been proven to be not only possible, but demonstrated to be potentially viable, is a very, very significant advance. As a doctor of the nervous system, I've always been frustrated with the idea that we can't fix the results of neurological diseases or injury. You know, we work on treating the underlying pathologies, but whatever deficits patients are left with are things that we have to live with. And that's very frustrating for us. And the idea that there are transformative technologies, discoveries that are taking place by really elite scientists such as Richard Anderson at Caltech and so on. The idea that we can take these technologies and bring them to human patients and see a way that our patients that we see every day can benefit from them is truly amazing.
My name is Heck. I am the medical director of the USC Comprehensive Epilepsy Program and the co-director of the Neurorestoration Center at USC. When one tries to bring an innovative and brand new type of device into humans, it takes a great deal of multidisciplinary approach. You need the scientists, such as Dr. Anderson's lab, but you also need clinicians who have experience in clinical research and working with humans. And so the challenge is to make sure that you're taking really good science that can be very groundbreaking and bringing it into humans in a very safe way. And that was where Dr. Liu and I played our biggest role. Anytime you take a patient to surgery, whether it's a clinical trial or simply to cure them for some sort of reason, you want to be very sure that they come out whole, just as whole as they were when they went in. And so when we brought Eric out of the operating room, our first concern is to make sure he was waking up and doing just fine, and of course he was. We are at a point in research in humans where we are making huge strides in overcoming a lot of neurologic disease. And these very important early clinical trials could open up and provide hope for patients with all sorts of neurologic problems such as stroke, brain injury, ALS, and even multiple sclerosis. And so it's not just about taking care of the subjects in our trial or the spinal cord injury population, but trying to move the needle for anyone with some sort of problem with their nervous system.